Now let us talk about the structures of tRNAs. Now the tRNAs are called the transfer RNAs. As we know, these tRNAs are actually the carrier for the amino acid sequences. It has an anticodon loop and it also have the attachment site. Now the tRNAs is exactly looks like the characteristic L-shaped structure, and the tRNA is possessing the tertiary structure in between among those all RNA structures. Okay, and among all those RNAs, tRNA possesses various types of what we can say the modified bases like pseudouridine, ribothymidine, or dihydrouridine, like this. Okay, so we have previously talked about a pseudouridine, where we have the structural difference in these regions. We have the ribothymidine, and we can also have the dihydrouridine, where we having the two hydrogen groups extra present there in those two positions. Okay, so the structures of tRNAs, as we know, have to be varied from one to another. So there are different 20 amino acids that you can find inside our body. So there we must need 20 different types of tRNAs for carrying those different 20 types of amino acids, and that's what exactly happening. So we are not only having one type of tRNA, we are having 20 different varieties of tRNA which are specified for attaching with a particular amino acid, which are specified for carrying a particular amino acid and carry them into the protein synthesis machinery, proteasome, into the protein synthesis complex. Okay, now so in this case what we can look, due to the presence of this slightly modified bases, they can have huge enormous they further increase the ability of making the hydrogen bonds. The further increase the making the capability of making bonds between the bases, and that actually helps them to make really strong structures. Now if we look at the picture here like that, the interactions fold between these different loops. So this is the secondary structure of this tRNA. If you consider this is the primary structure, all of the primary structure will have this linear strand only having the backbone and bases just coming out from the backbone. Now in this picture, if it folds, and it folds something like that to make the C prime end just getting more stretched, and 5 prime is shortened, and here we have four loops. At the below we are having the anticodon loop. In this anticodon loop we have bases, and this bases having the complementarity sequence for the proper codon that we can find in mRNA, where this tRNA is going to bind. And we have D loop in one side and the T size C loop in exactly the opposite side. Now this T size C loop, we are having the pseudouridine present there. That's why we call it the T size C loop, because we are having the thymine and pseudouridine and cytosine right after one another. That's why we call the T size C loop. And the opposite side we call them the D loop. We are having the GC here. We are having the adenine nucleotides here, adenine nucleotides, and they put together. So what is going on? This is the secondary structure which is pretty much stable, and we also have a short stretch of small loop which is called a variable loop, which is just in between the anticodon loop and T size C loop. Okay, now if we look at the structure, this is a structure that only can be made when we are having the stretches of nucleotide sequences that can have the freedom of making bonds between themselves more than the normal Arvind-Shargap pairing like AT and GC. So they can pair via GU pairing and all these types of pairings. And as a result of the presence of this pseudouridine and ribothymidine and dihydrouridine residues, they can further stretch this kind of capability to make another round of hydrogen bonding, another round of interaction, another round of hydrophobic stacking interaction between the flat faces of the bases to make a further round of complex structure, which we call a tertiary structure, as you can find in this picture. So this is the same picture which is denoting the which part of this region is start to interact with which part. So this G of this D loop is start to interact with this C, which is found in the variable loop. Now most of these interactions are being carried out from a D loop and T size C loop. So what we are generally looking at, just visualize this picture that this D loop is just coming out of this plane and it is going to interact with this T loop to make a folded structure, and this is the fold. This fold actually makes this structure the L-shaped and the characteristic L shape of this tRNA. Okay, so that's why this part is really important. We are finally folding this tRNA to make a tertiary structure. So everything is fine till that point. So due to the presence of this unusual bases, due to the presence of this modified nucleotide sequences, what we can do, what they can do, they can pair bonds with themselves, they can pair hydrogen bonds with themselves, they can also make the hydrophobic stacking interactions between themselves, and finally they can fold. And most of the time this D loop and T side C loops start to interact. They will fold to give the characteristic L shape to the tRNA structure, and that can make a really stable structure. Now we can find the structure something like that. Okay, so this is the secondary structure. It will fold something like this. So we can have different loops like this. Just consider this is the D loop. So we are having this D loop here, and we are having this T size C loop here. So this T size C loop and D loops are interacting, as we can see. So the D loop, which have to be present here, so it start to fold in this orientation, in this direction, as you can see here, and after the folding this structure becomes a structure like this. Okay, now at the end we are having the CCA tail, and this is the 3' end, and this is the 5' end, as you can see in this picture. Okay, so that's how this RNA structure is being made, and that's why the tertiary structure of RNA is being stabilized, because of not only the presence of the unusual type of base pairing, but also the presence of unusual bases. Okay, that's all, and I hope that's going to help you.